pints, so it'll be just a variable delivery, God help you. But just a few things in opening. First of all, I thought I might just open actually where I was going to finish off, which was exactly where you just touched on there, something positive, Katie, particularly around vaccinations. And I just want to start at last Saturday, this Saturday just gone. I spent a day in the Helix in Dublin City University, and it was where we ran the first vaccination programme in a vaccination centre for the 85-year-olds plus vaccinated by GPs, 100 GPs, 1,000 administrations of vaccine to the 85-year plus cohort population group. And it was probably one of the most inspiring days I've had in the last year since this has all started. It was just electric atmosphere. It was highly motivational, I found, and it certainly made up for many of the challenging days that we've all faced over the last year. Just to see the sheer joy on the families, on the people who are getting the vaccination administered themselves, people who are living in the community have had a really tough year. And there were just so many great stories on the day. People just wanted to come forward and thank the health service, thank us, thank everybody who works in the health service. And their gratitude was palpable. And I must say it's one of those days that made everything that's been so hard for the last year really worthwhile, because I know in terms of your own studies and medical science, it's key, but it just, I'm sure Cillian and others will talk about the advances, but for me, it just demonstrated, we've had a tough year, but we can see the chink of light. And there was many great stories. There was one person who was covered on RTE, Bridie was her name. She lives in Summerhill in the middle of Dublin city centre. And she was 103 years of age. And just talking to me on the day, telling me how it's giving her great confidence now to get back out again. So look, I just think it's things like that remind us the advances that's happened, number one. But secondly, there will be a way out of this very tough year we've all been through. It might take a bit longer, but there will be a way out. So that's where I was going to finish up. But now what I just want to give you a very quick background to myself, because I won't have met many of you previously, and then just walk through kind of learnings and lessons from the last year or so in a very brief period. So firstly, I'm with the HSE since May of 2019. I joined 6 months before this all happened. And it was never on the job spec, it was never on the interview questions. But it was about 6 months before. So I did have time before this all happened. Prior to that, I worked for 5 years as CEO of Fingal County Council. And for about 3.5 years before that, I worked as Chief Operations Officer when the new Department of Public and Expenditure and Reform was established back in 2011. I went in at the start of that. Myself and Robert Watt went in together, and it was that very tough period of time where you all remember the economic crash and what we were trying to do in terms of protecting the economy. Main career primarily for about for about a year and a half before that, I did some work for TROCRA, the Irish International Aid Agency. Many of you, I'm sure you're familiar with. And I worked and travelled all over the world with TROCRA, and again, a very refreshing period in my career. It was just fantastic. And then for a long time before that, probably for about 28-year career I had with Aircom. I was the executive director of their networks and technology and ICT and all of that. And going right back, just to help some of you in your own studies, I left school at 16 with an intercert. I'm sure none of you remember what an intercert was, but I did leave well before leaving cert. It was just the way things were. There just were no options for us to go on to college. In fact, there were no options for us to go on to myself and 6 siblings to go on to secondary education. But thankfully, like many, I got the chance for education later and did go on and do, obviously, leaving cert and obviously a degree and an MBA later on as well. So got one of those people who got a second chance in education, which really I grabbed, and it's proved well for me. Just, I suppose, homing in particularly, just looking back, there's a lot of milestones going around this week and over the next couple of weeks between when the first case was identified and the next few weeks about when lockdowns began, etc. But for us in the HSE, we and I go back to January the 27th, and I know Cillian does, when with the first day we had the National Crisis Management Team meeting in the HSE, because we were just beginning to see over that weekend cases emerging in Paris and Bordeaux from international travel from Wuhan. So first early indications of travel transmission into Europe. And we convened a team at that stage. And at that stage, to be frank, we weren't quite sure how to mobilise for all of this, because what we were seeing was the ECD risk was moderate and probably lower risk of it getting into Ireland. But, we convened a team at that stage and started looking at the basics, like how would our hospital system cope when, we know our hospitals are under pressure on good days. How do we mobilise more ICU beds? How do we mobilise more ventilators, PPE? And just give you a scale on the unknown that we faced into at that time. We would normally, on a normal year, spend 15 million euros in the HSE on PPE, which is a significant amount of money, 15 million euros. And we were grappling at the time, how much more would we need to spend? Would we need to double that? Hardly, we need to travel it. Would it need to be 100 million? And the actual figure we've spent on PPE this year is 1.1 billion euros. So nobody could predict the scale of what we were heading into. And that figure is a function of obviously volume of PPE we use, but also the price. What we were seeing in the early days, some of the basic masks, which were trading for about 37 cents under normal circumstances, in a very short period of time, went to €11.50 per mask. So give you a scale of the worldwide battle that was on for PPE. And we were securing orders, and actually many health systems came under pressure for their PPE and under criticism. But we punched well above our weight during that period, because we had our ambassadors across the world in Shanghai and all over Asia, working with the IDA, securing PPE for us. And I remember one night in particular, I described it recently as a modern day party, which was, in essence, we had secured a very big order for PPE from China. We probably paid about 5 times the multiple of what we really wanted to pay. But by the time that got to the airport for delivery and dispatch, other countries, I won't name them, but one other big country was bidding 5 times the multiple of what we had paid to try, get the supplier to give that order to them. So it just gives you a sense of the frenzy that was in that period. But really, just, I guess, the most important thing was what our staff, the frontline teams were feeling, and the public were feeling, which was quite a sense of being scared. And we all were, when we begin to see what was happening in Europe, when it gets to Bergamo, when it gets to Spain, Germany, France, it got really scary for everybody. The only thing I want to just say about that early period were a couple of things. And indeed, during that period, I think one thing that helped, 2 things I'll say, my experiences were, first of all, I was quite impressed, nearly blown away at times, by the whole of government response, how we knew very early in the health system that the whole of government were behind us. And there was one meeting in particular I reflect on, where we were in government buildings. I had just done a briefing about, we're probably going to have to scale up to another 6,000 beds. We're going to have to spend X amount on PPE, testing and tracing. And I was given all the kind of scaling up piece. And I remember at the end of that meeting, it was a very short meeting, and the Chief of Staff of the Defence Forces, Mark Mellett, came across to me and he said, you have the whole of the Defence Forces at your disposal. We're going to suspend all operations. Whatever you need, you have us. And I just thought it was an amazing moment in time for just, to see the power of the state coming together. And he had only finished saying that to me, and the Garda Commissioner, Drew Harris, came across and said, we're going to graduate all of our guards from Templemore, trainees from Templemore. You can have Templemore as a field hospital, and you also have air services for whatever you need. So I just thought that was a very powerful moment, and it held true to this day, how the whole of government departments, government agencies have come together to help the health system. And I think what benefited us from that approach were a couple of things. From the public's perspective, I'll just share a couple of feelings I have or judgments I've come to make. I think from the public's perspective, they heard during that early wave, during that first wave, a much more coherent voice from the health system. They didn't hear different interests coming out against each other. So traditionally in the health system, we're all used to whether it's representative bodies or trade unions or consultants or GPs having a pop off the HSE or the HSE at war with some of those. They actually heard a very strong, coherent voice between the department, between NEFID, between the HSE, between all of those consultants, the GPs. And I think that gave the public good confidence. And I can say that with a reasonable bit of knowledge because we did track public trust and confidence going throughout this pandemic and still do. And we did see, we all know, trust and confidence, particularly with the HSE, is never high in a given time. But certainly we did see it strengthen and grow throughout the whole period, which I think is something we all know is volatile, but we want to build on for the future. A couple of other final pieces just for those early phases for me is to say, one of the early messages I sent out with our own team working on this was that at best we were going to get 70% of our decisions right. And probably we were definitely going to get 30% of them wrong. But we should not be afraid of making wrong decisions to the greatest extent possible we can. But we should make sure we keep making decisions and make them timely, because the worst thing would be not to make decisions. And I can recall saying the same to government, saying the same to the cabinet committee, saying the same thing to the opposition leaders. You know, I did get quite a strange look when I always said that to them, but it was a kind of message that it's what happens when you're in a crisis. You have to make decisions quickly. And, you know, ideally we get a lot of them wrong, but some of them we won't, and some of them we didn't. But what I am happy for, and I've said this very often, the scale and pace of change that happened in that period of months in HSE is a scale that, you know, I've never seen so much change happen so quickly to be implemented by so many over such a short space of time. It was really inspiring to see that level of change implemented. And I think what worked for me was we got some level of decision making at the centre, but literally let the services get on with it. And I think that's a lesson for us, for the HSE, for the future, about empowering. I know it's often said that word, but it was about unlocking the system to do what was right and do it quickly and supporting them. I guess what has been different for me in the last, 6 months or 9 months or even a year has been that we have, I guess, implemented a lot of the reforms. I've often asked, I was asked in one radio interview recently, has this pandemic set back the reform of the health service? And I said then, which I passionately believe, no, it has actually accelerated a lot of the reforms of the health service. So we are now working in a much stronger way across the health system. We're working in a much more integrated way. And I know Cillian is here, and Cillian's going to be presenting to you in terms of labs, but mobilizing across the health system in a very different way, building capacity in 46 laboratories all across the country, building a testing and tracing facility with 47 swabbing centres across the country, building a capacity of 180,000 of swab and lab tests throughout Christmas. I think we saw the whole system working together. GPs working with us in a very new way, consultants working with us in a very new way, which is something we want to embed and instill for the future of the health service. Before I just come back to yourselves and the role that medical science has played overall, there were some kind of nice moments, I have to say, during it where, and I say trust and confidence was building up. There were some nice moments where I felt the public were getting behind us. When they needed us, when they needed to trust people, when they were scared, the health system were there. And there's a few kind of little moments that stand out in my mind, a few of them. But one was just people send me some, snippets and things on Twitter and social media, but there was one clip I remember back in May of last year where a community in South Dublin had come out to cut their kind of open green space shared between a small estate, and in between the green space, they had carved out with their lawnmowers, I presume, they had carved out "We Love HSE," and they had a drone footage sent across, and they sent it to me, and I just thought, I never thought I would see that. It's something very different. It was something really nice, and there were many little moments like that, and still today, all of our teams get some really nice messages from people. So I always say to everybody, Twitter is a bit of a false world. Don't take Twitter as real life. Very useful social media, but probably not absolutely reflective of what actually happens when you meet people in the street, or what really are people's experiences. So a few nice moments like that. What I just wanted to kind of, in the last few minutes, talk to you about is just in terms of the contribution of medical science, overall clinical and medical science, just to the pandemic. And I think, Katie, you spoke about at the start, who believed we would have a vaccine being rolled out on the scale that we have it, a number of vaccines being rolled out on the scale that we have. And that's probably a very clear demonstration of, the progress that has been made in medical science. But I think there's other ones as well. I think what was really pleasant, I found, for me throughout this whole process has been how we're using knowledge, how we're using data to inform us all the way, and hearing the public, through the likes of Cillian and the public health teams as well. A whole new language people never spoke before, epidemiological studies, talking about zero prevalence right now at the moment. But a whole language that people are now very familiar with. But as importantly, that we're using that knowledge, we're using that data to inform our plans and changing our plans in real time based on data, based on knowledge that emerges. Based on lab data from labs that's informing Cillian, informing us and HSE, based on modelling that can help us make certain projections about the disease, make certain connections between human behaviours and viral transmission. So, I think that's a whole new value add that's much more appreciated by society, much more appreciated by our communities in terms of how that helps the health system. Because I think we've managed to, during this awful phase, give the public a sense of the health system is not just about, the emergency department and our acute hospital systems. That's kind of the end point for people in terms of their healthcare. But it is about a whole value-added chain of knowledge, of data, of technology, of science that informs the health system. So I think we've got a much better base to build our connection with the public for the future, from my perspective. And the reason I say all of that is because one of the biggest challenges we have had, and I hope I gave you some examples during this, is about winning this public trust and confidence. But it's not just about winning the trust and confidence of the public. It's also about winning the trust and confidence of our funders, which are the public, but ultimately of government. And health system has always come under a lot of threat and criticism around funding. Should we put more money in? Where does it go? I think what's actually happened this year has been very positive for us in the health system and the health family overall, because I think government have built a stronger trust and confidence of the health system, our responsiveness to a pandemic. And that's the reason I think I can say that with some level of confidence is the level of funding that we've secured for next year, but also during the pandemic, because it's not easy to deal with the economic environment that government are dealing with at the minute and put almost 21% extra into the health budget for next year. It's gone from 17 billion euros to almost 21 billion euros. So that's a good signal that government understand and value investment in the health system based on our response to date. So I think, all of this knowledge, all of this data, all of this clinical science, all of the medical science, all our responses to the pandemic has strengthened the trust, I believe, of government in our delivery capacity and our delivery capability. So I guess, all of that for me is to say to yourselves is it plays a very key role. Medical and clinical science plays a really key role, not just in the pandemic, but in kind of the future direction of our health system and the future use of knowledge and data and technology and science about how we inform what's right for our health system for the future. So I think that's all put us on a much better footing. If I can just really come forward now to where we are at the moment, just overall in the health system, overall with the virus and overall with the health system in general, I think we're at a very positive phase because it's been a really tough few months since Christmas for everybody. And for our health system in general, it's been really tough for everybody. But we are seeing some really good positive trends, particularly over the last few weeks. Against all of the measures, we're seeing positive trends. We know it's equally at a very tricky phase because it does seem to have slowed a bit in terms of the progress. In terms of hospitalizations for me or people in ICU, it's going to remain extremely tight for the next few weeks. I know that because we know the pipeline of people we have sick in the hospital, the people who are receiving advanced respiratory support, will ultimately, a significant percentage of them end up in ICU. So unfortunately, we don't see the ICU numbers coming down to the extent we'd like just yet, because there's still a lot of very sick people in the hospital. And one journalist asked me recently, are you getting near to being normal? Is it getting near to being normal in the hospitals in particular? And my only response was, there's nothing normal about having almost 800 people being treated for the same illness in hospital at the same time. There's nothing normal about that. So that's the kind of levels that we're still at. That is tricky for us. But all that being said, I think we are in a, hopefully, a much better phase that can, as people see this light and hope in the vaccine, and I won't dwell on it too long, but just to give you a sense of it, to give you that sense of hope on the vaccine rollout, where we are unfortunately compared to our neighbours, the UK, who have played a stormer in terms of the vaccine rollout. Probably we would have benchmarked very well against them, against lots of things in terms of our response to the pandemic. But certainly in terms of the vaccine rollout, they did secure really good supplies. So, we are compared against the UK. Excuse me. But ultimately, we will see improved delivery, improved volumes coming through in the next quarter. What we are committed to in terms of volumes, what's being committed to us is 1.25 million by the end of March. And you begin to see that crank up now in the next few weeks. So we have been doing about 45,000 per week based on supply. We're administering everything we get within the same week. Last week, as in just gone, we completed 80,000 vaccinations. This week, it'll be close to 110,000. And it increases every week now. Ultimately, as we get into April, our deliveries are projected to be 1 million a month. So 250,000 a week. And being in that vaccination centre in the Helix on Saturday gave me a really good confidence insight into how we will run 40 of those centres all across the country, because that's what we'll be running in different sizes and scales. But we will be seeing, a kind of a much more mass rollout as these numbers improve, and we're recruiting the workforce right now for it. So for me, I think 2021 will be a better year. The one caution is always, every time we think we are at a level of predictability, the virus outmaneuvers us. When we thought we were going to get a break in August, it dragged us back. When we thought we were going to get a break at Christmas, it dragged us back. So we have to realise that we are living with a level of uncertainty, a level of unpredictability, and a level of virus that just seems to outmaneuver us every time we think we're on the right side of it. So that's a caution. But having said all of that, when you look at the positive signs and signals we're seeing from Israel, we knew, and many of you will have known, we knew that the virus will protect against sickness and hopefully hospitalization. We were unsure about transmission, but certainly the early indications from Israel are that a very significant defense, 89% defense against transmission, which, someday early data has to be peer reviewed, but I think that would give us all great confidence about what's being seen for the vaccines to date. So in closing for me, Katie, just apologies. I have to, I'm not avoiding your questions, and if I'm due to present at the Cabinet Committee for government's decision making tonight at 25 to 7. If I do get finished, I'll be more than happy to log back on and hopefully capture some of Louise and Cillian's presentation, but if not, even to answer some questions, I'll be glad to. But I'm in the hands of the government and the ministers on that one as they make their big decisions for tomorrow. So I hope that was useful, Katie, and I know you have very sexy slides and presenters coming up from Cillian and indeed Louise next, but I hope it's giving you some insight into what we've been dealing with and indeed the role that medical and clinical science has played.
Great, thank you so much. That was great. I think you've given us a really good indication of what your perspective on the pandemic was. And like you said, this was something that no one was expecting, but it is important to hold on to those little moments that give us hope. And I suppose the key to moving forward is communication and trust, as you said yourself. So thank you so much for joining us, and please do feel free to come back to us if you have time.
Hopefully, thank you.
Thank you so much.
Thank you.
So now we will have our second speaker. So we will hear from Dr. Cillian de Gascun.
Thanks, Katie. Thanks for the invitation to speak here this evening. Unfortunately, I'm not quite as well versed as Paul to speak for half an hour without slides. So I'm going to share my screen and rely on a crutch of PowerPoint. So fascinating talk from Paul, actually, and I could have listened to him for another half hour, because the last 12 months has been so I suppose incredibly challenging in many ways, but from a personal perspective as a virologist, it's been incredibly interesting as well, and perhaps not quite the experience that you necessarily want to have in the course of your career, but at the same time, it's a fascinating virus, and Paul touched on it at the end of his talk, the way the virus continues to evolve and change and make things challenging for us. And I suppose for me, having gone into virology, I suppose it's a privilege in some respects to see a new virus emerge, acquire, cross the species barrier, spread globally, and continue to sort of provide a challenge sort of 12 months on, and likely to become endemic over the coming years. So I am going to, I suppose, talk about the virus and I suppose some of the challenges we've had over the last 12 months in the context, I guess, about decision making, and Paul alluded to some of the stuff that I'll talk about in the context of trying to communicate uncertainty, I suppose, to the public in a situation where we're learning all the time, and we're acquiring new data and new science on an ongoing basis, and trying to interpret that so that we can, I suppose, develop policy and advise the members of the public as to how to behave in the context of what is, I suppose, and to use an overused word, has really been unprecedented. We've obviously seen the pandemic of 2009 with influenza, but really that obviously doesn't compare to what we've seen over the last year. So I think this is really certainly a first in a lifetime experience for the majority of us, and hopefully will be once in a lifetime, but I'm not overly convinced that it will be. So I people will obviously, we've just gone past, I suppose, the coming up to the anniversary for lots of things in Ireland, but people will be familiar with the background of the virus, which I won't go into in great detail. I suppose what's key for me here is the fact that the sequence of the virus was shared so quickly. So the Chinese reported the novel coronavirus as the causative agent of this outbreak of a cluster of pneumonia of unknown etiology on the 7th of January. And then the viral genome sequence was available on the 10th. And what that allowed us to do, I suppose I'm using us generously, what it allowed the scientific community to do was to develop a diagnostic test very quickly, because PCR is obviously very specific. You require specific primers and probes. And if you have the sequence available, you're able to develop a test that can then look for that particular pathogen. And we started testing here on February 7th. So that's really just about a month after the first sequences were made available, and the first diagnostic assay was published. The methods were published in around the 28th or 29th of January. So a really impressive, I suppose, scientific contribution to allow us to diagnose it. I suppose one of the key things for us at this stage is that we still don't know where this virus came from. And again, from a personal perspective, as a virologist, that's something I'm curious about. And the reason being that if we want to, I suppose, try and prevent future events of this nature, it helps to figure out where this one came from. There's always a risk of fighting the last war, but at the same time, information is knowledge. So if we look at this slide, this is the sequence of the SARS coronavirus type 2 aligned with all of the other coronaviruses that we know to date. And up at the very top, you can see SARS coronavirus type 2 there in red. And just below that, you can see a bat coronavirus that is its closest relative that we know about at this stage. And then below that again in cyan, you can see a lot of coronaviruses that have been isolated from pangolins. And that led to a lot of speculation that pangolins might have been an intermediate host for this virus, in the same way as civet cats would have been an intermediate host for the original SARS coronavirus back in 2002 and 2003. But we don't believe that to be the case now. And then scattered through the rest of that phylogenetic tree on the left-hand side in green, you can see our other seasonal human coronaviruses. So we believe that this virus came from bats, but we don't really know how it crossed the species barrier, and whether there was an intermediate host. And obviously the WHO has been over in China recently trying to carry out an investigation. But one of the, at its most basic level, if we don't know where the virus came from, we actually don't know if there's an ongoing risk. We don't know whether cross-species transmission is still ongoing, or whether this was a once-off event that has now stopped. Looking back at our other seasonal coronaviruses, they've all come from either bats or from a rodent natural reservoir. Some of them have had intermediate animal hosts, some of them we presume most of them have, but some of them we know, some of them we don't know. There's a very nice review article that came out at the end of 2019, so unfortunately just came out before SARS coronavirus type 2, but it's a really nice review of what we know about the human coronaviruses to date. So just to flag, so what I'm highlighting here, this is just a schematic of the genome of coronaviruses up at the top, and we're expanding out or blowing up the spike protein and some of the other structural proteins. And what I'm highlighting here in the middle with the green dot is just there are 4 different genera for coronaviruses, and SARS coronavirus type 2, along with the original SARS coronavirus and MERS, fits into the beta coronavirus family. And I suppose the structure here will touch what I'm highlighting here is that this is obviously just a schematic, but the thing I want to highlight or draw attention to is just the spike glycoprotein on the outside. This is what the virus uses to get into our cells, and obviously it's been the target for all of the vaccine candidates that we have to date. And that's the viruses, or the vaccines rather, have obviously been incredibly efficacious, and they've been developed so quickly. But one of the challenges that I'll touch on later on, obviously, is the issue of emerging variants and what happens when that spike protein changes, because obviously viruses continue to evolve and they change all the time, and coronaviruses are no different. This is just looking at the life cycle of SARS-CoV-2. Through the spike protein, it binds to 2 receptors on the cell surface, 2 is the primary one, and then there's another transmembrane serine protease that it binds to, and then the virus sort of comes into the cell. The RNA is released, then we get replication, we get generation of the viral proteins and transcription of the virus genome, and then each virus gives rise to, on the right hand side there, new virus particles budding out from the cell. So in the course of this pathway, the virus is going to make mistakes, in essence, as it replicates its RNA, and that leads to evolution and change. So we know from studies over the last 12 months is that SARS coronavirus type 2 acquires mutations at a rate of about 1 to 2 per month, and that's led to the emergence of lots of different lineages, and obviously the novel variants that are of concern at the moment. So I'll touch on those later on. I'm not going to go into the schematic, I suppose, but one of the things when this virus first arrived, we obviously assumed it was going to be very much a respiratory virus based on its mode of transmission, and obviously there was significant respiratory pathology, which I suppose came to light very early on. And this just highlights, again, from a nice review article, just highlights what's going on in the lungs in people with sort of significant respiratory distress syndrome associated with SARS coronavirus type 2. But obviously what we've learned over the last 12 months, and one of the challenges I think has been trying to figure out the extent of disease that this virus can cause. For us, we're a new host for this pathogen, for this virus, so it is still finding its feet. Obviously, it's been an incredibly long year for us, but from a virus perspective, we're still a new host. It's still getting used to us, and it's still figuring out how best to transmit and how best to cause disease. Although it's not really necessarily in the virus's best interest to cause significant disease, what the virus is trying to do is really, it wants to find a host, it wants to replicate, and then it needs to be able to get out of that host to find another host. And that's why respiratory viruses can be so efficient. So if you think about it in this context, if this virus were to get into an individual and not replicate as much in the airways and went down to cause a hepatitis, for example, then those particular clades or those particular subtypes may not transmit terribly readily and may not sort of survive. But at the moment, this virus is really finding its way in a new host, and it's causing significant extrapulmonary disease as well as respiratory problems. So I think when we look back to the start of all of this, and we knew that this was a second or a third coronavirus, I suppose, in the 21st century. I suppose the question we obviously tried to learn from what we had seen previously with the original SARS coronavirus and indeed MERS coronavirus from 2012. And SARS coronavirus from 2002, 2003, that caused about 8,000 cases worldwide with a case fatality rate of about 10%. But the kinetics were very different. So obviously we were trying to learn from that, but also we had to keep an open mind as to what might be new for this new virus. So we know that SARS coronavirus type 2 has a higher reproductive number. People will be familiar with all of these terms now. Again, as Paul has alluded to, people have learned a new vocabulary over the last 12 months. We know that SARS coronavirus type 2 binds more strongly to the ACE2 receptor and is more efficient at getting into host cells. And it also has a greater affinity for the upper respiratory tract, which again leads to infection of the upper conducting airways more easily. Viral load kinetics also explain some of the differences. So this is, I suppose, what we're looking at here. And again, what we had to try and look at last year as things evolved was this virus going to be contained like SARS coronavirus type 1, broadly speaking, or was it going to become a global pandemic? So unfortunately, obviously, it's gone on to become a pandemic. And one of the reasons that has happened as well is because of the viral load dynamics and the duration of infectiousness. So again, we were learning all of these things, I suppose, in real time last year, and from a public health perspective, I suppose from an effort perspective, trying to base advice on emerging evidence. And I suppose the first comprehensive report we had was from the World Health Organization that went on a mission to China to try and figure out collate the information from the Chinese experience and then disseminate that through countries globally, so we could see what the issues were. So if we look back to SARS coronavirus type 1, the highest viral loads were typically detected in the upper respiratory tract in the second week of illness. So people weren't terribly infectious early on. But obviously SARS coronavirus type 2 is very different, and almost sort of the exact opposite. So the peak viral load is observed really at the time of onset of symptoms, or in the first week, or even before the onset of symptoms. And that makes it very difficult to control. So the original SARS coronavirus was really controlled with isolation and quarantine, purely because people weren't terribly infectious in that first week. So if you identified people with symptoms, they weren't transmitting infection before the onset of symptoms. So it made it quite straightforward in some respects to control, notwithstanding the challenges of controlling any new pathogen. But if we look at SARS coronavirus type 2, it's been completely different. And we also know that people who are severely unwell or who are immunocompromised may have a sort of a prolonged course of viral shedding. But we know from early on that in the majority of patients, SARS coronavirus type 2 RNA is detectable by PCR in the upper respiratory tract for a couple of days prior to the onset of symptoms and for about 7 to 12 days afterwards. So that was the other feature, I suppose, of SARS coronavirus type 2 that made it difficult and more challenging to contain, was this idea of pre-symptomatic transmission, so people transmitting the virus before the onset of symptoms. And again, that impacted on our public health guidance. People will remember back to the idea of the topic of wearing of masks. And while it was always recommended very early on for people with symptoms, there was a sense the guidance changed when we realized that pre-symptomatic transmission and that people could be genuinely asymptomatic, because that wasn't a big concern from the initial report that the WHO convened out of China, and it probably reflects the virus sort of evolving over time as well. But obviously the guidance then changed around mask wearing when we realized that pre-symptomatic and asymptomatic transmission were genuine concerns. This is a nice sort of overview, I suppose, of the data that emerged, and while it's a single slide now 12 months into this pandemic, but it takes an awful lot of time and an awful lot of data and work, I suppose, for people around the world to try and collate information and to publish data. And I suppose we've all become familiar with the concept of preprints over the course of the last 12 months. They haven't all turned out to be correct or accurate, but it's been a very positive, I think it's reflected very well on the scientific community that people are willing to share their data so quickly. And also a lot of journals have made their SARS coronavirus type 2 material free to the user as well, so that data are being shared in real time insofar as is possible. Again, I mentioned this earlier, so I won't spend too much time on it, but we still don't know where this virus came from. We know there are hundreds of coronavirus circulating in bats. Obviously SARS and MERS came from bats. But until we find that progenitor virus for SARS coronavirus type 2, or sort of the ancestral virus for SARS coronavirus type 2, we can't be definitive as to where it might have come from. So its most closely related virus to SARS-CoV-2 is a coronavirus that was identified in a bat species in Yunnan province of China in 2013. But what's interesting about that virus is that although it's 96% identical across its genome, the receptor binding domain within the spike protein is quite different. And that receptor binding domain is more similar to the coronaviruses from Malayan pangolins, and that was what's led to original speculation. But the receptor binding domain is key for us in the context of trying to figure out where this virus may have come from, and also why SARS coronavirus type 2 has been so successful in humans. So the receptor binding domain is the most variable part of the genome, and there are 6 amino acids in the receptor binding domain that are shown to be critical for binding to the ACE2 receptors. So that these sort of amino acids in essence then determine the host range of this whole family of viruses. So interestingly enough, SARS coronavirus type 1 and type 2 are different in 5 of those 6 key amino acids, but 6 out of 6 are the same in pangolins. So we also know from studies that have been carried out that SARS coronavirus type 2 can bind with high affinity to in humans, obviously ferrets, pigs, cats, and non-human primates. And what this suggests to us is that SARS coronavirus type 2 had time to evolve in the presence of a human or a human-like ACE2 for natural selection. So to touch on that, why has SARS coronavirus type 2 been so successful in humans and managed to transmit so readily? So there's a polybase cleavage site insertion at the junction of S1 and S2. These are 2 subunits of the spike protein, which is not observed in other lineage B beta coronaviruses. And this allows for effective cleavage by furin and other proteases and plays a role in determining viral infectivity and range. So furin is abundant in the respiratory tract, but is also found throughout the body and other organs. And the cleavage of the S-glycoprotein typically facilitates viral infection, whether it be through enhancing cell-to-cell fusion, as in SARS coronavirus type 1, or enhancing the ability of coronaviruses from one species to infect another species. And acquisition of a polybasic cleavage site in influenza viruses also enhances viral pathogenicity. So that insertion is really of interest to us, but we don't know where that came from. So this is just a schematic of the spike protein, and then we're blowing up the part of the receptor binding domain here. This is aligning the human SARS coronavirus type 2 and align sort of most similar viruses, so the bat or ATG13, pangolin, and then the original SARS coronavirus from 2002, 2003. And what you can see with the purple arrows, that there's this polybasic cleavage site that is present in human SARS coronavirus type 2, but is not present in any of the other similar viruses. So we don't know whether that happened during human-to-human transmission or whether it happened before the virus crossed the species barrier. And that's one of the questions, I suppose, that we'll hopefully get an answer to over the coming months or years. The other thing that's identified on this slide are O-linked glycan residues. And what these are sort of, I suppose, residues are amino acids where polysaccharides or sugars or sugar moieties can be added. And this suggests that this is sort of a method that viruses often use when they are evolving to conceal, I suppose, epitopes or antigenic epitopes from an immune system. So this sort of blue, so it's fuzzy material, are all sort of polysaccharides linked or sort of attached to the spike protein into various residues. So again, what those residues suggest to us again is that this virus has had an opportunity to evolve in the presence of an immune system. It doesn't necessarily tell us which immune system, but it does give us some information. So again, just to wrap up on this piece of the talk as to where the virus came from. So did it cross the species barrier fully formed? So there was obviously the link. This means that the virus just evolved in an animal host before jumping into humans. So obviously we had the early association with the live animal market in Wuhan. We've also talked about the pangolin coronaviruses, but we haven't identified this polybasic cleavage site in any of the viruses sampled to date. Now, obviously, we haven't surveilled all animal coronaviruses, but at the same time, the fact that we haven't seen it would suggest that it may have arisen in humans after the cross-species transmission. And equally, we haven't identified a SARS coronavirus type 2 progenitor that we can pin down at this point. So the other option is that the ancestral or the progenitor virus crossed the species barrier into humans and then evolved subsequently in humans. So it may have acquired the genomic features that I've talked about above through adaptation during undetected human-to-human transmission. And then once those features were acquired and the virus became more readily transmissible in humans, the pandemic would kick off. So we have seen the receptor binding domain configuration with those 6 amino acids in nature, in pangolins. But as I said, we haven't seen this polybasic cleavage site insertion. So speculation at the moment is that this occurred during human-to-human transmission, and it could have happened if there had been many sort of prior zoonotic events with short chains of human-to-human transmission, as has been seen with MERS coronavirus. And it's possible now that unfortunately lots of samples are probably gone at this stage, but if there were banked human samples from the second half of 2019, we might be able to see if there was any evidence of cryptic spread prior to the pandemic kicking off. So in general terms, we feel that option 2 is probably most likely. And I feel, again, as a virologist, that understanding the origin of this pandemic is important because it might give us information and allow us to, we may not be able to definitively prevent future zoonotic events, but it might give us some information that we can include into a surveillance program. It might highlight things that we should be looking for. But as I said, at this point, we don't know where or how the cross-species transmission occurred. One of the questions that always comes up, and obviously it's relevant in the context of the variants, and I'll touch on those later on, is are there different types of SARS coronavirus type 2? And yes, there are. And unfortunately, there are 3 different naming systems that we have at the moment from GIS8, from NextStrain, and then from Pangolin, recommended or described or developed by Rambo and colleagues. So these are, I suppose, the different lineages, or these are the different terms that you'll hear when people are talking about different types of SARS coronavirus type 2. There's lots of diversity in Ireland. This is just an example from a slide. This is a couple of months old now at this stage. Up on the top left-hand side, this is the pangolin nomenclature. You can see lots of different lineages. And then down at the bottom, you can see what pangolin starts for there. So the phylogenetic assignment of named global outbreak lineages. And what pangolin is trying to do is, I suppose, identify lineages that are relevant in a particular region at a particular point in time. They can, the naming may change over time, and it's not intended to give you a comprehensive history of where the virus came from, but it is useful to describe, I suppose, outbreaks and monitor evolution in a particular region. So if we look at Ireland's pandemic over the last 12 months from a sequence, so these are all whole genome sequences. In Ireland, we've sequenced over 2,000 whole genomes at this point in time. So just over around 1% of the pandemic over the last 12 months. So if you look at over on the left-hand side, you'll see B1 lineage, because that's associated with the European lineage that's associated with the Italian outbreak back in the early part of the pandemic. And you can see then that we've moved through. Now, that doesn't necessarily mean that we imported it directly from Italy, although we probably did have cases directly from Italy, but that lineage spread throughout Europe. So you could have introductions from other countries. Over the course of the next sort of 6 to 9 months, you can see we had other lineages such as B1174, B136, and B1258.2. Some of those are linked to the UK, some of those are linked actually to the Middle East. And then you can see around August, sort of between July and August of 2020, you can see that B1177 appeared. So that's what has commonly been called now the Spanish variant. But we are trying to, I suppose, encourage people to describe these variants as being first described in a country rather than being named by the title, because obviously people know that Spanish influenza had nothing to do with Spain. So B177 is that variant that emerged during the course of the summer, and we believe will sort of disseminated or spread throughout Europe in association with travel, in essence, as people thought we could open up again and start to live life a little bit more normally. And then you can see over on the right-hand side of the graph, you can see there's plenty of diversity there, but obviously B177 accounted for probably about 75% or maybe 85% of cases at one point in time. And then you can see that B117 started to emerge in or around December and now accounts for about 90% of cases. So that's the variant that was first reported in the UK. And interestingly enough, while B1177 appears to be purely based on a confounder effect, it doesn't appear to have any genetic characteristics that would make it more transmissible. It just spread in large numbers. B117 does have genetic features and mutations that appear to make it more transmissible, and I'll touch on some of those later on. But these data are from NextStrain, I should add, so anybody who's sequencing in Ireland uploads all of their sequences. I should acknowledge the Irish Coronavirus Sequencing Consortium that have done a lot of the sequencing in Ireland, and they're all of the sequences that are shared with GISAID, which is a global initiative for the sharing of influenza data. And anybody can go on to NextStrain and see these pictures and monitor them over time. So I suppose one of the other challenges we've had over the last 12 months is trying to, I suppose, communicate the science to people and figure out what the risks are. So this is a respiratory virus primarily, and it's spread by respiratory droplets. And the risk of acquiring infection depends on a variety of factors, such as contact patterns, your environment, the infectiousness of the host, or the stage of disease that the host is at, socioeconomic factors such as overcrowding, and then preventive measures. So we know that most transmission occurs through close range contact, defined as 15 minutes face-to-face within 2 meters. But we also know that spread is especially efficient within households and through the gatherings of family and friends. And we've seen that over the last year. Household secondary attack rates are a really good measure, I suppose, of the inherent transmissibility of a virus. And that ranged from 4% to 35%, depending on the studies you looked at over the course of the last year. But equally, sleeping in the same room, being the spouse of an infected person, all of those things increase risk. And the risk of infection substantially increased in enclosed environments compared with outdoor settings. And obviously there have been reports of airborne transmission across a distance of greater than 2 meters. So I just wanted to touch on this because it's one of the things, I suppose, that has attracted quite a lot of attention. Droplet spread versus airborne spread. So droplets are typically can come in all different sizes. Heavy wet droplets fall to the ground very quickly, and then smaller, lighter droplets can stay airborne for a prolonged period of time, and that can be minutes and it can be hours. And is it possible for people to acquire infection from those airborne droplets? Of course it is. Generally speaking, however, if we look at the relative contributions of droplet spread and close contact spread versus the possibility of airborne spread, we'd be fairly confident that droplets account for the majority of the patterns that we're seeing. Is it possible that airborne spread can occur in certain settings? Absolutely. Do we believe it's a significant driver of the pandemic? Probably not, purely because the methods that we introduced last year wouldn't have been sufficient to control it. But I think it is important to realize that all of these things are a spectrum, and they're also dependent on environmental factors. So these are why ventilation is so important. Humidity obviously has an impact, temperature has an impact, because that has an impact on how the virus survives and how long the virus can survive in the environment. Fomite sort of contaminated surfaces, obviously regular cleaning and hand hygiene are so important, because we know the virus can remain viable for up to days on smooth surfaces. But again, it's a key issue, and it's really important for hand hygiene and to regularly clean surfaces. But it's probably more of a significant factor in facilities where there are communal areas and environmental contamination and sort of many high touch surfaces. So again, that's one of the things that feeds into our guidance. But also, as we saw over last year, trying to keep the guidance up to date and ensure that you're not confusing people with the message was really important and quite challenging. Just to touch on some of the transmission questions that came up. So as I said, household transmission gives you a really nice indication as to how transmissible a virus is in its natural setting, if you like. And there's a couple of studies that showed nicely that the secondary attack rate for SARS coronavirus type 2 in households was in or around 20%. And that compares with, say, chickenpox, for example, which is up at 90%. But what we've seen with the lineage B117 first reported in the UK is that this secondary attack rate in household settings has increased from about 20% to about 30%. And that sort of fits with the 50% increased transmissibility that was first reported by colleagues in the UK. If we look at the idea of sort of asymptomatic and pre-symptomatic infections, again, there was speculation, I suppose, in the early stages that the majority of people might be asymptomatic, and that would make it very challenging. But with a number of studies over the last year, we realized that probably the majority of people, sort of 70 to 80% of people who become infected will develop symptoms of some shape or form throughout the course of the infection. And obviously we were learning about symptoms over the course of the last year as well. Initially, we thought it was very much a respiratory virus, pure and simple. But then obviously we learned about the loss of taste and the loss of smell. And I think one of the challenges over the course of a pandemic with the new virus is figuring out what's genuinely associated with the new pathogen, because you're also trying to figure out what the mechanism of causing that symptom might be. And then the last slide on this transmission is just around about asymptomatic transmission and how much they contribute to the pandemic. So obviously asymptomatic infections can occur, but how many people are those individuals transmitting the infection to? And again, while it's certainly possible and certainly does occur, it's likely that symptomatic individuals, and that's sort of intuitive, it kind of makes sense that if people are coughing or are shedding lots of virus, then they're probably more likely, if they're short of breath and breathing heavily and expelling sort of lots of particles, respiratory particles, they are more likely to play a greater role in the spread of infection than people who don't have symptoms. But as I said, symptomatic infection can certainly transmit the infection. And again, that was one of the things, I suppose, from a learning perspective last year, was also the question. There are two questions I suppose we always need to ask ourselves: can something happen, and is it a major driver of the pandemic? And I think then trying to communicate that to the general public, I think, was one of the difficulties or one of the challenges that we faced, purely because communication of complex sort of issues where even sort of the scientific evidence doesn't necessarily always agree can be quite challenging. So just to run, I'm not going to spend a huge amount of time on the testing, but just to go through, just to sort of highlight the process there. People will be familiar with it at this stage. I'm sure many people have been tested in the audience. we're still using primarily the nasopharyngeal or nasal swab. It goes into sample transport medium, which in non-pandemic times, we actually like to put the virus into viral transport medium so it keeps it alive so we can grow it in the lab, and we would do that for influenza quite regularly. But obviously, given the categorization of SARS coronavirus type 2, we actually tried to get it into a lysis buffer, which inactivates the virus at the point of testing. And then that sample comes to the lab and we extract the RNA. So just coming back to one of the points Paul was making about capacity and about laboratory testing in the 48 labs. So I think we're up over 3 million tests at this point in the country, and the National Virus Reference Laboratory in a normal influenza season. So our influenza season typically runs from the start of October till the end of April. So we know that we'll get our influenza activity at some stage during that period of time. And what we would do in an average influenza season, so between October and April in the NVRL for influenza, is about 15,000 tests over that period of time. And obviously, now with the HSE and with the support from the HSE and all the investment that they've put in place over the last 12 months, like we're routinely doing 25, 30,000 tests a day across the laboratory network. So that includes the hospitals and hospital laboratories, it includes ourselves here, and then also support from private providers that the HSE had to bring on stream to scale up the capacity to allow us to be able to deliver the testing that we needed. So again, what Paul alluded to, the fact that we didn't necessarily have this infrastructure in place before the pandemic, but certainly we're hopefully coming out of the pandemic in a better place than we were going into it. So it hasn't necessarily delayed investment and expansion, it's enhanced it, if anything. Broadly speaking, we're still using reverse transcriptase PCR over on the left-hand side of the slide. There's been some work done in isothermal amplification, which people will be familiar with from a LAMP perspective, or LAMP technology. That has been used in certain settings, and there's some research still ongoing. It's never really made it through into, say, the high throughput laboratory settings, but it's certainly useful in the context of, say, either near patient testing or either low and middle income countries, or in non-traditional laboratory settings, because it's a little bit more flexible. CRISPR-based detection we haven't really used. I'm not going to go into that. I suppose the one thing in the context of testing I just wanted to touch on again, because it's one of the challenges over the challenging areas over the last 6 to 9 months, because there's been so high profile, is around antigen testing. And I think there's probably a perception out there that I'm anti-antigen testing, which is not true. But it's funny, the HSE has done a huge amount of work in evaluating these antigen tests, and there's been a huge amount of attention around them in the media. And Dr. Mary Keoghan and Dr. Rain Doherty have chaired a group that has looked at this, and Professor Cara Martin from Trinity has done a huge amount of work on evaluating antigen tests. And unfortunately, they just didn't perform as well as the manufacturers said that they would. Now, they are validated and they're being rolled out, and they're available in Ireland now for use in symptomatic individuals. This is just a sort of a snapshot of some guidance from the WHO that came out at the end of last year, and just highlights the challenge of using a test in a particular setting. So we know that they work quite well in people with symptoms. They certainly work less well on people who don't have symptoms. But that doesn't necessarily mean you can't use them in that setting, but you need to compensate for that lack of sensitivity, either through frequency of testing or through increasing the amount of testing that you're doing. But certainly from the point of view, and also if you don't have access to PCR, then certainly antigen testing in symptomatic individuals or in asymptomatic individuals, if they're close contacts, is better than nothing. But again, thanks to the investment and the work that the HSE has done over the last year, we do have significant PCR capacity at the moment, and hopefully we won't get to a stage again as we did over Christmas where we had to suspend the testing of close contacts. Just for a reference purpose, I suppose these are ECDC guidance, so just so people may not be familiar or may not understand, I suppose, how we decide who we're testing and why we're testing. And this is just a sort of a brief overview of what the principles for testing are. So you're obviously trying to control transmission, you're trying to monitor your incidence and trends over time, you're trying to protect your healthcare settings and social care settings. So that's being done with, say, serial testing in our residential care facilities, and obviously testing people as they come into hospital. You're obviously trying to look at clusters and outbreaks quite quickly. And then you're trying to prevent reintroduction of the virus into regions or countries with sustained control of the virus. And that really comes back to airports and other ports of entry. And certainly, as we've seen with variants in recent weeks and months, you really want to introduce some element of testing with travel to fully protect your borders. Obviously, we're recommending against all non-essential travel at this stage, but we know that a certain number of people are traveling. So the government has introduced a requirement for people not to have a not detected PCR test before travel, and then obviously their quarantine on arrival. So that travel issue, I suppose, has also been a challenge over the last 12 months in the context of getting the balance between what's best for public health, but also what's right for the economy. So I won't go into the vaccines in great detail, purely because I'm not a vaccinologist, but just, I suppose, to touch on them because they've been so, it's been such a good news story. I certainly didn't think that 6 months ago that we would have 3 vaccines on Ireland and being rolled out within that time frame. And it is a phenomenal, again, achievement of the scientific community, and shows what can be achieved in a short period of time when it's needed for for emergency services. And I just wanted to select one graph just to show, and people will have seen variations of this from a variety of different studies. This is from Pfizer-BioNTech study, and it just goes to show in the red line, the flat red line at the bottom, when the vaccine kicks in compared to the placebo group, you can see the number of cases or the cumulative incidence in the placebo group over time, and these are really striking results. And as I said, they've been replicated across the majority of other vaccine candidates out there, and it's really impressive to see, and does bode well for the future. But unfortunately, I suppose there's always a challenge that I'll come to in a second with variants. But I think this has been a really nice schematic from Ian Mackay, who's an Australian virologist, and he had this out a number of months back now at this stage, because I think there's a tendency for us, and there has been a tendency to look for a single silver bullet, whether that's testing and tracing, whether it's quarantine, whether it's isolation, or whether it's ventilation, or whether it's masks. And I think this just is a really nice schematic showing that no single intervention is going to be perfect, is going to be 100% perfect. That's not the way these things work. But what we can do is, if we combine all of these things together from a personal responsibility perspective, and then at a societal level, and we add vaccines onto that, each intervention has imperfections, but multiple layers, and the more layers we have will improve our success. And I think that's a really nice sort of schematic just to look at. And then just to finish off, because again, this is very, very topical, and I suppose as a virologist, it's something that I'm interested in. So there are 3 variants. I'll spend, just to go through the principle, I'll touch on the UK one first. But so an analysis of routinely available genomic data in Kent was undertaken. So what happened in southeast of the UK was that they had fairly significant restrictions in place, and they noticed the case numbers were coming down across the country, except in Kent, and they couldn't figure out why. So they did an epi investigation to figure out, was it associated with a particular activity or particular population group or a particular location, and it wasn't. So they looked at the sequence data, and they noticed that a large cluster of genomically similar viruses over a period of time. And then they did some studies, and they realized that this variant at the time was spreading more quickly and more rapidly than other lineages that were around at that point. So what's interesting, so what is unique about this lineage? So it's called lineage B117. It has an unusually large number of genetic changes, particularly in spike protein, and 3 of those mutations have potential biological effects that have been described. So people have heard of probably 501Y, so it's a key contact residue, and appears to increase binding affinity with human ACE2. There's a spike deletion that has been reported a couple of times, and again may increase transmissibility. And then there's another mutation immediately adjacent to the furin cleavage site, which is a location of biological significance. So the most unusual and concerning mutation in that cluster was 501Y. And I suppose when we talk about an unusually large number of genetic changes, we still don't know how this virus emerged. As I said to you earlier on, we know that SARS coronavirus type 2 accumulates mutations at a fairly steady rate, typically unless there's an unusual situation, and that's the unusual situation. There's been speculation that this virus may have emerged in the setting of a persistent infection. So there wasn't significant immune activity to eliminate viral replication, but there was sufficient immune pressure to put an evolutionary pressure on the virus to cause it to adapt or to change. So 501Y appears to increase transmissibility, as that affects the receptor binding affinity of the spike protein, and it's possible that mutation alone or in combination with the deletion is enhancing the transmissibility. So that mutation has been found in other animal adapted viruses, but it's also present in the other variants of concern that have been reported in South Africa and Brazil. And I suppose what's interesting about this amino acid change is that it's emerged differently, or emerged independently rather, I should say, in a number of different lineages in a number of different settings. So it's obviously a configuration, and it's obviously an amino acid change that the virus likes. So what this amino acid is doing to the virus is that it is making it fitter. It's making it more fit from a replicative perspective, and it's giving it a fitness advantage, which is why in the course of 2 months here in Ireland, it's gone from being less than 2% to over 90%. And the concern is that this will be the new baseline, because the virus will continue to evolve. So the next virus that, whatever virus displaces 501Y is really going to have to be at least as transmissible, if not more transmissible. Thankfully, from an antigenicity perspective, 501Y doesn't appear to have any impact on the vaccines that we have rolling out here, so that's good news. So the key thing for us is still to get the vaccine rolled out as quickly and efficiently as possible. And this is just a phylogenetic tree of this. Again, this is a few weeks old now at this stage, but it was just looking at the 501Y mutation sequences that were on that were in GISAID, and you can see over on the right hand side there in blue we've got the South African variant, and then in purple we've got the UK variant, and then you can see a couple of green sequences around there. So they're Irish sequences that possess the 501Y mutation, and there's a number of the fact that those sequences aren't clustering together would suggest that even at that stage there was a number of different introductions of the virus into Ireland. So it wasn't like a single introduction, but obviously we had travel at that time. So the nerve tag was an advisory group for respiratory viruses that was set up in the UK. And so what they provided a summary of the B117 lineage, and they suggested it had a growth rate that was about 70% higher than other variants. They reckoned that it increased the reproductive value by a factor between 0.4 and 0.9. They looked at CT values to look at viral load and suggested that there was a decrease in CT value, which means an increase in viral load. And certainly that's been shown through the S dropout work from Thermo Fisher as well. But in all, they were happy to report that this virus was more transmissible, in part because it was better at infecting cells, and in part because of it led to an increased viral load and more infectious. And I suppose this is just a reminder of what we've seen in Ireland over the last couple of months with B117, and the pattern that we have seen and the growth rate that we've seen in Ireland would fit very, very nicely with an increased transmissibility and an increased growth rate of between 50 and 70%. Just to touch briefly on the South African one. So, as I said, B117 from the UK doesn't appear to have any impact on vaccine effectiveness. What's concerning about B1351 is that this has other mutations in addition to the 501Y, which may well impact vaccine effectiveness. So if we look at South Africa's pandemic, again, looking over on the left-hand side, you can see B1, that's the lineage that we saw from Italy and in Europe. And then you can see significant diversity there in the middle of that graph. But over on the right-hand side, you can see how B1351 just took off probably around September time, probably emerged late August, September, and then it's just become completely dominant in South Africa over that time period. So the reason we're worried about it is because in addition to 501Y, it also has a 484K, which confers resistance to class 2 neutralizing antibodies, and 417N, which has an impact on neutralizing class 1 neutralizing antibodies. So all of these these are reasons for concern, because we know that these antibodies, obviously, our neutralizing antibodies, bind to these sites. So in simple terms, the protein that we now have in all of our vaccines has changed in this virus. So if were this virus to become globally dominant, then this would be a concern for vaccine programs. Now, obviously in Ireland at the moment, our key role is to prevent introduction of this virus into Ireland, and certainly if it arrives, to contain it. We've got about 14 cases at this point in time. But it's unlikely to displace B117 at this stage. But at the same time, we want to roll out vaccine as quickly as we can. And then looking at the variant first reported in Brazil, lineage P1. So this emerged in Manaus in the Amazonas province in December of last year, January of this year. And I suppose this one was most concerning because it emerged at a time when there was a presumed very high level of immunity. So this region of Brazil was severely affected back in April of 2020. And there was a reported seroprevalence based on seroprevalence studies of in or around 70+%, suggesting that a significant proportion of the population had been exposed to the infection back in March, April, May of last year. But the fact that this variant was able to kick off again around December, January raises questions as to how that happened. So obviously either the seroprevalence rates were overreported, potentially there was waning immunity, or else it's due to the virus itself. Perhaps this virus is more transmissible, or perhaps it's evading an immune response. Again, just to show you the schematic of what's happening or what has happened in Brazil. And again, down at the bottom right-hand side, you can see how P1 has emerged sort of in or around probably November time when it was first reported, and then has kicked off. Now it's hard to know at this stage whether it's plateauing at this point or whether it's going to continue to increase, but certainly it is a concern. And if we look at the amino acid changes in these variants, so we've UK at the top, South Africa in the middle, and Brazil at the bottom, you can see that there are common patterns across the board. So 501Y is in all of these, 484K is in Brazil and South Africa, 417 position changes in Brazil and South Africa. So Brazil, very much like South Africa, is probably equally transmissible based on the 501Y, and then may well be capable of evading vaccine responses based on the 484K and the 417T. So this is the type of thing I suppose we expect to see with viruses. And obviously you'll all be familiar with the fact that we change our influenza vaccine every year. So coronaviruses aren't influenza viruses. They don't mutate at the same rate. But what's interesting about these variants, if you look at the number of mutations that are listed there, there are more mutations there than we would expect to see at this point in the pandemic. So if we want to try and figure out where these variants are emerging from, and why they are, what evolutionary pressure they've been exposed to, then that might allow us, if we can learn about these, that might allow us to ensure that the same set of situations or that the scenario doesn't arise again where more variants will emerge, where the viruses will continue to evolve and continue to change, and that's going to be a challenge for us. But I think the positive note is that the public health measures that we have in place are capable of controlling all of these variants. But we do know that with the B117 in Ireland at this stage, that the restrictions we have in place will have to last for longer, and it will be more difficult to suppress purely because it is more transmissible. And I think that's just one of the challenges for people, is that what we achieved last year in April and May was really impressive. And the problem is the same level of effort this year isn't going to have the same result. So if people feel that it's harder this year, it's because it is harder this year, and communicating that to people and trying to keep them with us and keep them engaged and keep them following the public health advice, I think is one of the challenges that we will face over the coming months as we roll out vaccination and hopefully bring a level of suppression to the virus. So just to finish off then, when we see these new variants emerge and the questions we're asking, we're looking at transmissibility, we're looking at severity of disease, and there's a signal now from the UK about severity of disease with B117, and obviously we're looking at vaccine effectiveness. So it really highlights the role for surveillance and whole genome sequencing, and that's not only country specific. So we need to look for outbreak, because the problem is if you target specific countries, viruses don't recognize borders. So if we just target people from Brazil or South Africa, then we may miss people coming in from South America, we may miss people coming in from Southern Africa. So the other thing that our public health colleagues would always do is they would flag outbreaks or clusters with unusual features. And then one of the things we'll be doing over the coming months now is looking at infections in vaccine recipients to look for genuine sort of vaccine escape mutants. And then in the future, we'll either be looking at updated vaccines. We know that some of the manufacturers have already started to do that. And the other question is, can we look at a universal vaccine that will target a more conserved region of SARS coronavirus type 2, so therefore the virus can't actually mutate at that point, or if it mutates at that point, there's a fitness cost to the virus rather than a fitness benefit. And maybe we need to look at expanding it out from just the spike protein, because as I said, in simple terms, the spike protein that was there last year is now different, and that's going to be a challenge for our vaccines in the coming months and years. So thank you very much, and I'll stop sharing there.
Great, thank you so much. That was really informative. I think something you highlighted that was very important is that while we do have the answers to an awful lot of the questions, there are still many questions that need to be answered, and the only way we can do this is through study. And I suppose, as they say, knowledge is power, so it's very important that we'll be able to make collaborative efforts to find the answers to these questions. So thank you. And now I will hand you all over to Miss Louise Barry from Cork University Hospital. You're just on mute, Louise, sorry.
Okay, right, can you hear me there, Katie?
Yeah, that's perfect.
Okay, great. So look, first of all, I just want to thank the UCC Biomedical Science Society for an invitation to speak here this evening and share, I suppose, our experience in the Department of Clinical Microbiology in Cork University Hospital, I suppose, around implementing a SARS-CoV diagnostic service while maintaining a full microbiology service. I suppose to explore, what the role of the medical scientist was, the challenges that we encountered along the way, and I suppose increasing capacity to support community testing and where we are today. No. Yeah, sorry now. So look, I suppose if we just reflect for a moment, and it's obvious that we've come a long way in the last 12 months, where our first case of SARS-CoV in Ireland was on the 17th of February. And I suppose it's just staggering what we've achieved. Only 1 month previously, on the January 17th, WHO gave guidance on laboratory testing in relation to the novel coronavirus. The pandemic was declared then on the March the 11th, and on March 16th, the clear and unambiguous message from the WHO was to test, and this is how we're best equipped to deal with the coronavirus pandemic. The call to test and increase COVID testing capacity has placed medical scientists front and centre to both the diagnosis and management of patients, but also in the curtailment and the spread of the virus. So in CUH alone during 2020, from a service that we implemented on the 5th of March, and I suppose a service that this time last year we're only thinking or planning to implement, we achieved 97,133 tests. I suppose this was superimposed on an already busy diagnostic microbiology service, that we were already beyond capacity with annual increases of workload of the magnitude of 4 to 5%. Testing, as both Paul and Cillian have alluded to, is happening on a national scale across the length and the breadth of the country in many hospital laboratories, 31 acute labs and 17 private labs, and being supported by 5 community testing labs. I suppose it's important to acknowledge that this hasn't happened in silo. We have had the support of the HSE, the Department of Health, the HPS procurement, and the National Clinical Pathology Programme. So just a little background to Cork University Hospital for anybody who may not be familiar. CUH is a model for tertiary referral centre and a university teaching hospital. CUH is unique in that it has 40 surgical and medical specialities. It's also a tertiary referral centre for the full southern region and also serves a super region of 1.1 million, covering Limerick, Kerry, Tipperary, Waterford and Kilkenny. Together with Mallow and Bantry hospitals, we all compose the Cork University Hospital Group. Average presentations that we see in our ED department in 2019 was just short of 70,000. We have 32,000 inpatient discharges in 2019, and we have a staff of 3,488. So the Department of Clinical Microbiology itself, we're the largest microbiology laboratory in the region. We provide a consultant-delivered diagnostic microbiology service across a full range of bacteriology, infectious disease, serology, and TB. And we provide this to our own hospital, the maternity hospital, and both Mallow and Bantry who are in our group. We provide referred services and specialist work to both the Mercy University Hospital and University Hospital Kerry. We also provide extensive GP service to the region, and about 50% of our workload actually is generated from the GP and from the community. We're an accredited laboratory. We work to the ISO 15189 2012 standard. We're committed to providing the highest quality of patient care, and this is right through the patient journey. We have a dynamic multidisciplinary team within the department composing of consultant microbiologists, specialist regs, medical scientists, MLA, and support staff, which is composed of clerical and housekeeping. We currently have 1.5 consultant microbiologists, which is well below what a hospital of our complexity and size should have, and we have a staff of about 40 medical scientists. It's important to note that the microbiology service composed of, I suppose, the consultant microbiologist, the scientific and the infection prevention control team have at this stage provided a 7/7 diagnostic SARS-CoV testing service for COH and the hospitals in our group for the last year. So I suppose I just wanted to explore the role of the medical scientists, and I suppose I'll do this over the next number of slides. And I suppose how the role of the medical scientist has diversified and evolved over the last number of years, and how this diversification has been fundamental in shaping the laboratories where we now work today. I suppose traditionally when we consider laboratory medicine and medical science, we tend to measure our output by, I suppose in microbiology, the number of swabs we do or the number of urines that we process. But I suppose if we look beyond the benches, and the added skills that the medical scientist has honed over the last number of years, it has enabled us to adapt quickly, and this is exactly what is required in a pandemic. So I've put health and safety at the top because I suppose staff safety and well-being has always been really important, and this is, I suppose, more important in delivering a service throughout the pandemic. Laboratory medicine and microbiology, we provide a 24/7 service, 365 days. We didn't have, I suppose, the luxury of being able to shut our doors. We had to continue providing this service. And we've always been leaders in laboratory medicine and microbiology when it comes to health and safety. Following HPSC guidance, it was necessary to risk assess our processes to ensure that we had safe work practices. And also we had to do this again when we had to make changes to our safety statement and safety procedures on the back of the work safety protocol, which were implemented to ensure that we were maintaining a safe work environment. Maintaining staff safety was always a priority throughout the pandemic. And I suppose prior to the pandemic, it's true to say that most people wouldn't have known what 2 meters was. The labs that we work in, you know, we're in a building which dates back to 42 years and the labs are in the current footprint. So I suppose they're not really fit for purpose and social distancing was hard to achieve. So then superimpose, I suppose, a very busy service in on top of an already service that's at capacity. This is a challenge. So for the first wave, we decided to split into 2 teams. I suppose we spread ourselves out over the course of the day, and this ensured that we both got the work done, and I suppose we kept our staff safe. And this was only available to do, I suppose, staff were willing to engage with this and were happy to do it. And also in the initial phase, we had curtailment of services, both from the GP and because there was curtailment of services within the hospital itself. If we look at our quality system, we have a quality management system which is well embedded within the department, and we have a robust change management process which is administered locally by our quality coordinator, and it's overseen and managed by the laboratory quality manager. So I suppose this means when it comes to any change management or introducing any new test into the laboratory, this can happen smoothly without any interruption to service. The change management process covers everything from when you have your kit, looking at the kit insert, is the kit CE IVD mark, the verification plan, the verification process itself, the staff training surrounding this, and I suppose the quality control, EQA. So to put this into perspective for our own laboratory, within 4 weeks of our first SARS-CoV positive on the 5th of March, we were running on 3 extraction platforms with 2 different amplification kits. So we were already in 5 verifications 4 weeks in. And to year's end, we would have had double this number of verifications happening in the laboratory to sustain our service. IT is very important for any new service that you're implementing. The test needs to be ordered and it needs to be reported. So this involves new test codes, lab comments. How will the lab result be reported? Is it detected, not detected? Is the reporting manual or is it interfaced? And what will the report look like on our own laboratory information system, but also on foreign systems such as GP messaging, the maternity system? And how will this data be captured for our notifiable diseases through CIDR? All these changes need to be made in the test environment before being released into the live. So project management. We're fortunate to have the ability to utilise the expertise of our blood sciences project manager to help with a number of projects involved in SARS-CoV testing. And this is most notably with regard to in providing community testing on site here within the department. And the role of the PM would have been involved in planning, organisation and seeing the schedule to completion. All staff in the department are involved in staff training, and medical scientists are fully involved as we're a teaching hospital for both biomedical science students and medical students. There's a systematic approach to training in the department, which is both procedural and bench-based, which marries with the educational requirements of the staff member who's being trained. As a department, we continue to train because I suppose we had to, because on the back of providing a new service, we had to take new staff into the department. Across the whole MDT team, we had 33% new staff in 2020. And if we look at the medical scientists and the MLAs alone, we had 25% new staff. I suppose we had to continue with training, but we had to adjust how we did it. We've always been committed, I suppose, to research in the department also. And we had 5 MSc students undertaking their research part of their MScs at the time that the pandemic hit the department. So it was through the commitment of supervisors and the staff on the ground that these projects were seen to completion during 2020. So point of care. So this is laboratory service which takes place outside the realms of the lab, and it's in the bedside essentially. So we've a newly appointed point of care team in the laboratory medicine, which is originated in our biochemistry department. And the expertise and assistance of this team will be utilised for the provision of COVID-19 testing as necessary outside of the laboratory setting. So if we look to our molecular expertise that we had within the department, we've long established molecular expertise in the field of molecular diagnostics. And certainly a lot of this initial work would have been pioneered with Dr. Bridget Lucy, who you'd all be familiar with from CIT. So she would have established a routine diagnostic service for enteric pathogens back in 2008. I suppose as a department, we'd previously responded to the swine flu pandemic back in 2009. And molecular testing is now very much part of our routine diagnostic service across, I suppose, a wide range of bacteria, viruses, and the unculturable organisms such as chlamydia. And molecular diagnostics have proven invaluable for an acute hospital being able to respond to outbreaks such as norovirus and MDROs, multi-drug resistance, which are, I suppose, very important in the acute setting and the hospital setting. So these skills, so we were no stranger, I suppose the other important point to point out here, we were no stranger, I suppose, in the realm of molecular, being able to do less with more. We've investigated practical solutions to technology. So to make them cost effective to implement, I suppose, into a routine diagnostic service. So we had previously looked at pooling for CPEs. And I suppose this is a tool that could have or may have the potential to be useful in the context of a pandemic where reagents and consumables were very in short supply. So if we look at surveillance, microbiology laboratories have well-established laboratory-based surveillance systems. They've been developed for the systematic ongoing collection, analysis and interpretation of data and the timely communication of this for action to public health and I suppose control. CIDR is computerized infectious disease reporting of communicable diseases to public health. And I suppose this is an established system for reporting from the lab to public health, and this would have formed the basis for track and trace, which was very important during the pandemic. Additionally, locally, surveillance in conjunction with our hospital IT have developed and implemented a data management system which delivers data to the National Integrated Information System for testing and contact tracing. And also, we would also provide up-to-date trend analysis of COVID-19 positivity data for local trend analysis to, I suppose, the Department of Public Health, to our hospital CEO, and to group and to occupational health. So I suppose looking at the testing and our journey, I suppose, to mass testing. So I've used hashtag flatten the curve here. I suppose this is part of a public health strategy to slow down the spread of the virus and build on healthcare capacity and mass testing. I won't go into, I suppose, the different forms of testing, because this has already been covered by Cillian. But to say that we were using nucleic acid tests which detect the presence of viral RNA using real-time reverse transcription polymerase chain reaction. So I suppose in the microbiology department, when we were planning to introduce SARS-CoV testing back on the 20th of February, we had two considerations: what extraction platforms were available to us, and the fact that we had an open system for our light cycler, what amplification kits could be used. So I suppose we didn't necessarily want to reinvent the wheel, because this was timeliness of a service was very important. So we looked to our current suppliers and we chose the kit that was currently being used by the NVRL, the Altona. At the time, this was a non-CE IVD mark kit. So I suppose it all kicked off for us on the 5th of March, where we had our first community acquired case. So with this patient, he was now currently in our ITU, and there was no travel history, and he was very unwell. So on that same day, our kit came in, which enabled us to start a SARS-CoV testing service. And I suppose this gave us the impetus to I suppose, get a move on. While we did this all very quickly and hastily, there was no compromising on the quality. So the verification was started on the evening of the 5th of the 3rd, and we would have tested in tandem with the NVRL over that full weekend, and we would have continued to reflex our first positives to the NVRL. And by Monday, the 9th of March, the NVRL were satisfied that we could be a testing site for SARS-CoV, because up until that juncture, the only testing site in Ireland was the NVRL themselves. So we were the first lab outside of the NVRL and the first acute laboratory to provide this service. So now that we had a SARS-CoV testing service, we were able to offer this to our own hospital and the hospitals in our group. But we also tried to support other hospitals in the region, namely University Hospital Kerry, Mercy University Hospital, and the Bon Secours in those initial stages. And we had a team of 6 medical scientists. And I suppose this was provided 7/7, and it was a very demanding service. Almost immediately, we were hit by global reagent shortages that affected our extraction system, MagnaPure 24. We had to look internally within laboratory medicine as to what we could do and how we could mitigate against these global shortages. So we had a shared molecular extraction platform, the MPLC2, which was shared throughout laboratory medicine. So following training and verification on the company training on this system, we went live on this on the 13th of the 3rd. And this gave us, I suppose, additional capacity, and I suppose it gave us redundancy, although the extraction process itself was somewhat slower. I suppose the fact that it was an older system as well, we probably had a more secure consumable and reagent supply line for extraction. I suppose due to the skilled nature of the service that we were now providing, and it was always a worry in the back of our minds, what if anything happened to the 6 medical scientists who were providing this service? And we made the decision there and then to expand the service to hematology, who were familiar with the MPLC2 extraction system. So now we could have 2 independent teams who were able to provide the service. So my next slide is on hashtag in this together. So the global reagent shortages really became relentless by mid-March. And it definitely became a daily struggle, if not an hourly struggle, to sustain our existing service we were literally hand to mouth with our suppliers, and we were borrowing from other laboratories who had similar platforms. So for the MP24, we were borrowing from Limerick, Galway and St. Vincent's. So the reason I've put the hashtag in this together, so this is about staying connected, which is very important and was very important to us and proved to be a lifeline. Through connection and networks, we reached out to pharma and academic partners looking for any potential assistance that they could offer us. So this also gave us an opportunity as well to be leaders and innovators. So following a lead from Dr. Bridget Lucy, we went and visited animal health laboratories in Bandon, who they were reaching out to us to, I suppose, offer us some capacity on some of their extraction analyzers. While this wouldn't have been suitable to us as an acute laboratory service, we did have the opportunity to look at their extraction processes. And these would be extraction systems that wouldn't be normally used in the clinical system. But I suppose, as Paul and Cillian have both said, that you have to think outside the box, and we made a decision very quickly. We needed to sustain our service, and we needed to increase capacity. And I suppose we needed a guarantee of a supply chain. So we placed an order. Literally, we went and saw the system on the 19th of March. We placed an order for an IndyMag on the same day. The instruments arrived in the department on the 24th of March. They were implemented into the department. We had online training on the 25th of March, and we were up and running by the 27th of March. And we had a secure supply chain with these reagents on the 30th of March. I suppose another area that was very much suffering due to the increased testing nationally and internationally was the lysis buffer reagent to extract the viral DNA. So the microbiology department in CUH, we worked with other scientists across academia and biopharma to pool our expertise, our technical know-how, and I suppose the chemicals that they had available to them from their research laboratories to make this critical reagents, the lysis buffer. So CUH was a verification site for a number of these, I suppose, trial lysis buffers, which were submitted from Eli Lilly, UCC and Chagas. We went on to share our findings of this manufactured product that we had made, I suppose, globally, really, in case anybody else is running into the same difficulty. This was shared in an international publication. The lysis buffer went from here to being made on a national scale by NIBRT and was subsequently distributed by the HSE. I suppose while we had a secure enough extraction platform, we were still running into difficulties with our amplification kits. And I suppose we began to think outside the box a little bit, on two fronts. What if we could forgo the extraction process? And I suppose a kit that we'd be familiar in the laboratories offered by Ceresep is the Enteric Bio, where they can detect RNA and DNA directly from the fecal samples themselves. So I suppose we would have collaborated with Ceresep in developing and seeing if we could look to forgo the extraction process. While this, I suppose, R&D didn't go quite as well. It was successful, but it didn't go quite as well. It wasn't quite as sensitive as our extraction processes. Ceresep did produce an amplification kit called the Recipe Bio, which CUH ended up being, I suppose, one of the national testing sites for this. This kit was released for use into the Irish and the international market on the 24th of April as a research use only, and subsequently was CE-IVD marked on the 15th of May 2020. And this was a kit that, I suppose, combined with our IndyMag, offered us capacity, but also gave us a quicker extraction platform, because this was the times for the amplification and the extraction were much reduced. So we had quite a sustainable service. It's important to mention as well the National Clinical Programme for Pathology, which came on board mid-March. And this was a welcome relief, I suppose, certainly to ourselves and I suppose to all the laboratories around the country who were trying to sustain the SARS-CoV testing service. And this gave us, I suppose, we were all competing with each other at this time. So I suppose there was a planned program for what platforms the people would use and what amplification kits they would use going forward. And it also gave us buying power, so that we were trying to compete in a global market where all reagents were short. So if we then look to our own lab and building capacity and how we did it, I suppose while we were happy enough with our current system of the IndyMag Recipe Bio, it was working well. And I suppose we were sort of removed from the crisis of the global reagent shortages for the main part. However, sustaining our service was becoming quite challenging. We now had a team of 7 medical scientists who were providing this service over 7. After an initial dip in our own workload in the microbiology department of 20%, I suppose this was again beginning to restore to normal. And I suppose with the national shortage of medical scientists, we just couldn't get any more medical scientists into the department. So this current system was really not sustainable. And I suppose the big part that it was lacking, it just wasn't automated. It was semi-automated with a lot of manual pipetting steps and manual interpretation. So in the numbers that we were doing, and if we were to increase capacity, it just wasn't feasible. So as the HSE, in conjunction with the National Clinical Programme for Pathology, looked to increase capacity nationally, as a laboratory, we were offered the opportunity to have an automated large-scale batch analyzer called the Cobas 6800, which we see in the picture here, which would support community testing. I suppose the key decisions for us, the department around it, has the benefit of being automated with automatic result reporting. We were also continuing to provide our service, which was superimposed on our microbiology laboratory. We were working out of the same safety cabinets that we were doing our specimen setup in for our main microbiology, and this was challenging, so we needed a new space. As I said, the medical scientists, they just were nowhere to be had nationally and locally, so we could look at skill mix. And also, if we were looking to the winter ahead, we didn't know what it would hold, so it was important to plan for testing for flu. So I suppose this system offered this with dual testing from the one swab. So from when we accepted or decided to go with the Cobas 6800 in COH, it was signed off by HPS procurement on the 28th of 4th. We're really on a tight time schedule to get this up and running within the department. So an MDT multidisciplinary team was convened, and I suppose there was 3 strands to it. I suppose we had the build itself, we had the IT connectivity, and the technical issues of introducing the assay into the department. So we had a number of stakeholders. We had HBS Estates, procurement, and in COH itself, we had laboratory medicine, we had IT, and we obviously had the provider, we had Roche. I suppose we initially had to find a place to put it. Unfortunately, we couldn't accommodate it within laboratory medicine, but we needed to find a site close to the labs themselves. So the microbiology department had an original molecular diagnostic unit, which was across in our maintenance yard. So I suppose this was decided that the site was suitable and was close enough to the laboratory to be suitable. So we spent a lot of time with Roche, and I suppose designing a suitable floor plan. I suppose we didn't just have the instrument itself, which was a large capacity analyzer, but we had also all the ancillary things which come with supporting a service. So we had safety cabinets, benching, computers, we had a server, and we had storage for reagents and consumables. So all this was signed off on mid-May, I think it was around the 21st of May. So we first started with our modular build. This was installed over a weekend on the 22nd to the 24th of May. It was an 85 square meter build. And Estates then had 1 week to get this modular building up to being able to accept the Cobas 6800 that you see in the picture to be built on site. So there was groundworks involved. And from once the instrument came on site on the Tuesday after the June weekend, we then had a 6-week build to build the instrument, commission, train the staff, verify the instrument and the IT connectivity. We went live with this instrument on the 17th of the 7th, 2020. So this is the Cobas 68 testing platform. And I suppose from concept on the 28th of April, this was a really quick turnaround time for, I suppose, a build with the build instrumentation to going live of only 3 months. So if we look to the providing community testing, we had always been providing some part of community testing. This was for priority, so where we had healthcare workers or maybe where we had symptomatic individuals in the community. However, to enter these into our lab information system, and to meet the track and trace requirement. I suppose it was challenging because there was an awful lot of manual entry involved. There's an awful lot of, I suppose, you had telephone numbers, you had Swift QIDs, and I suppose there was an awful lot of training involved for our clerical staff. Another issue that we were experiencing was because we were now covering a large area, not all GPs were set up on our lab information systems. So to ensure that we could report these in real time to the GP messaging systems, we needed a lot of IT support where the GPs would have been set up so that we could report out through the GP messaging systems. So once we went live with the COVID-68 analyzer and once we were selected as a pilot for community testing, it became obvious that we needed to circumvent both of these issues. So we needed to introduce SwiftQ, which is a patient appointment system, which is used to order the SARS-CoV testing in the community. And this now needed to be interfaced to our lab information system. So we had the full connectivity from ordering to result reporting and track and trace the community. So providing this community testing, I suppose, has been very challenging. I suppose we have a new stakeholder that we have to take account of, and the accurate, timely reporting of these results is constant, and I suppose we're constantly being audited for KPIs. I suppose we're fortunate in that we can flex up and down with our own work. So if we have a busy day or we have PM on the instrument, we don't have to take as much work from the community, and I suppose we're satisfied with that, as are the community. So there's no increase in turnaround for the patient at the end waiting for the SARS-CoV test. And I suppose since we introduced the COVID-6800 on site, we've had a 65% increase in the sample numbers with the community testing place in-house. So I suppose looking to the challenge of the third wave and what it meant for us. So winter is traditionally a very busy time for the microbiology department due to seasonal influenza. And I suppose fortunately this year, even though we had planned for it, there was no influenza, which is due to the measures introduced to suppress the COVID-19 virus. So annually we would support our batch multiplex respiratory testing with the Gene Expert, which is a rapid flu testing. And we would have introduced our SARS-CoV testing in a rapid capacity on this back on the 15th to the 4th. There's always a necessity to complement your batch testing with rapid testing because not everybody can wait for the time for a batch analyzer to provide a result, and there'll always be patient cohorts that need to have their results turn around quickly. So in the third wave, locally, we were seeing a substantial increase in positive test numbers, both within the community and within our own patients being admitted to the hospital, and unfortunately, some of them being admitted to our ITU. And I suppose this was reflective of the national picture that we've all seen over the last month. So I suppose we unfortunately would have had a number of COVID-related outbreaks in COH, and this has placed a huge burden on us, both the hospital itself and the associated teams, the microbiology medical team, infection prevention control team, and occupational health. So the microbiology team are very important in this, in that staff, it's important that they report, I suppose, the positive results cases, and they're discussed at a clinical level, and I suppose just to ensure appropriate placement within the hospital itself. Annually, we have about 70,000 ED admissions that I previously said, and in order to maintain patient flow throughout the hospital, the microbiology department had to increase its rapid testing capacity that it could provide to ED. So to do this, we had to I suppose spread ourselves over a number of rapid testing platforms, notably the film array and the ePlex. So we went live with the film array on the 15th of the 12th, and we also followed suit with the ePlex on the 20th of 12th. This gave us sufficient capacity over the three testing platforms, and unfortunately we couldn't consolidate on one, which we would like to, because I suppose they all have different times to give off a result, because I suppose of the global pandemic that we're in. Additionally, in the last 2 weeks, we've taken the decision to introduce an overnight rapid testing service for our SARS-CoV to our ED department to assist with patient placement and assist with patient flow in the hospital. So COVID-19, what are the next steps? So I suppose COVID-19 has certainly provided an impetus for introducing point of care. And we're currently in the process of introducing molecular tests, the LIAT, in our ED department. We're working with the laboratory medicine point of care team, ED and IT, and testing will be performed in ED by the ED staff for immediate action. And the results, the output will be captured by the laboratory information system for reporting to CIDR and follow up by public health for track and trace purposes. This training is going to be rolled out in the coming weeks with ED and the company involved. As Cillian has already alluded to, the usefulness of rapid antigen detection testing kits in an acute setting is questionable. I suppose we're aware that they have reduced sensitivity and ideally they should be carried out on symptomatic individuals, but there's no denying that they may have some utility in an outbreak situation. I suppose we're all very concerned about the new variants and they're a real concern. And I suppose we would have to question whether it's time that we need to reflex our positives to determine the appropriate epidemiology. These multiplexes are now readily available for the 3 known variants, the UK, the South African and the Brazilian. This could happen quickly and easily in a laboratory level who are already providing testing. Sequencing. ECDC recommend sequencing 5 to 10% of our positive isolates. And I suppose we would have to wonder whether we can do this locally and use the expertise of laboratory medicine, namely in histopathology, or whether this should be done in a more centralized setting. So I fear retesting of positives. This is always a challenge. I suppose we would really have to question whether we should be retesting positive patients from whence they were within the 12-week period from when they're initially diagnosed. I suppose this can be to do with inconsistency around results, around limits of detection of various testing platforms. And it may be more of a clinical decision, you know, when somebody has recovered from COVID-19 rather than a laboratory-based test. Unique patient identifier, the COVID-19 has certainly highlighted this as a requirement really. And I suppose there would be margin trying to capitalize on this and on, I suppose, the advances that have been made. We regularly see a scenario in the acute setting where a patient is tested in the community and they then deteriorate and become unwell and have to be admitted in through our ED department. And we've no visibility on their COVID test that has already been tested in the community setting. So this is an important thing. I suppose it has so many implications for so many reasons, you know, vaccine rollout. But I suppose for microbiology, we're conscious of multi-drug resistant organisms, and when somebody is entering the hospital setting, they don't park their bugs at the door when they're being admitted. So look, I suppose it's true to say it's certainly been a challenging year for us as a department, but individually as a department, we've achieved so much, and in the most challenging of years, we've maintained a full microbiology diagnostic service during the global pandemic. We were the first acute hospital to implement a SARS-CoV testing services. This was accredited last October as part of our INAB annual assessment, and we were the first site to support community testing and in an acute setting. Medical scientists as a group were only small in the context of healthcare. We number about 2,000, but collectively we've achieved so much. We've implemented and sustained a quality assured SARS-CoV diagnostic testing service on a national level, the length and breadth of the country. And I suppose the real success has been our staff, the medical scientists, how we have used our expertise. We've thought outside the box, we've been flexible and adaptable, and we've been unwavering in our quest to fight this pandemic. We've certainly been more than test. Thanks very much.
Great, thank you so much, Louise. I think you really nicely highlighted how the medical scientists are more than just, you know, testers conducting tests each day. There's a lot of work that goes into implementing a new test and assay the quality, everything that goes with that. And from spending some time in the laboratory over the summer, I think it is safe to say, you know, there's a lot of expertise, and it's just a matter of utilizing it as best we can. So thank you so much.